Hello again. I'm doing another video post, this time on Chapter 2 of Deep Nutrition by Catherine Shanahan. In the first chapter, I talked to you guys about what epigenetics was, a little bit of background on the author, and how food can kind of regulate your genes. Today I'm going to be talking about Chapter 2, which is titled The Intelligent Gene, Epigenetics and the Language of DNA. Basically, this is going to talk about how, complex the system is and how food really is the language of how our genes change. To begin with, I guess we should know some basics about DNA so you understand how the whole process works. Basically, in every single one of our cells, it has a nucleus, and inside that nucleus are 46 chromosomes. And those 46 chromosomes each one of them has 300 million pairs of genetic letters. So basically, these genetic letters are made up of nucleic acids, and there's 4 basic letters. There's A, G, T, and C. And how the language of DNA is told, basically, is the pattern of these letters. And before, it was thought that to change the pattern, you had to do entire letter substitutions. So, say the pattern was A, G, T, C. So to change the pattern, you could go A, G, C, and then T. But now it's being found out that instead of changing entire letters, you can change the DNA simply by having these temporary markers on them, or these epigenetic tags is what they're being called. And throughout our lifetime, they can either detach or accumulate. And those tags are basically how your genes read the rest of your DNA. What's crazy about our DNA is that only 2% of it is your genes, those static things that control your eye color, hair color, your height, kind of things like that. But 98% of them, of your DNA, used to be called junk DNA. Not because it was junk or unuseful, it was just that we didn't understand what it was used for. But now we're beginning to understand that all of that junk DNA is really what can change in your DNA. So 98% of your DNA is able to change and shift according to what you do in your life, basically. The reason that we know that our genes can change and shift was that there was a twin study done. It took 2 sets of twins, one's at the age of 3 and one's at the age of 50, and they basically colored their DNA and to see what would change over a course of a year. And the 3-year-olds, their DNA was almost exactly the same, which follows the assumption that identical twins would have identical genes and DNA. Now, the twins that were 50, when they colored their DNA, it was completely different. There was so many different things in there. So basically, that showed that life experiences changed DNA, and that they didn't, grow, and their genes didn't change in the same fashion just because they were identical twins. It was their life experiences and what they did in their life, whether it was eating, exercise, smoking, not smoking, things like that. It changed their DNA, and basically their genetic makeup was completely different. Scientists are identifying numerous techniques by which 2 sets of identical DNA can be coerced into functioning dissimilarly. This is on page 26. So far, the processes identified include bookmarking, imprinting, gene silencing, X chromosome inactivation, position effect, reprogramming, transvection, maternal effects, histone modification, and paramutation. And many of these epigenetic regulatory processes involve tagging sections of DNA with markers that govern how often a gene uncoils and unzips. Once exposed, a gene is receptive to enzymes that translate it into protein. If unexposed, it remains dormant, and the protein it codes for doesn't get expressed. So basically, that's like, let's say you have a gene that's used for bone building. That gene would have 2 tags on it, one for vitamin D and one for calcium. So, in that way, if you have a lot of vitamin D and a lot of calcium, that gene would get information that says, we have exactly what we need, vitamin D and calcium, so it's going to uncoil, basically, and then the protein would be open for the environment, and now, because you have enough vitamin D and calcium, the bone-building gene is active, is turned on, and then you'll start building more bones. But let's say you don't have vitamin D and calcium, that gene remains closed and dormant, and you don't build as much bone over your lifetime. So that's one example of how those tags can work. One of the biggest things that we found is that the tags can fall off, and that way a gene forgets what it's supposed to do. So over time, if, let's say, in your family history, your ancestors never ate enough calcium and vitamin D. So basically, that gene, those tags, those vitamin D and calcium tags, will fall off, and then that gene forgets how to build bone, basically, because the tags are no longer attached to the gene. But that doesn't mean that just because you have a history or you're going to develop osteoporosis, that's what the doctors tell you, doesn't mean that you can't change that. Because tags are just made from simple nutrients. That's what's crazy. It's made just from simple nutrients like minerals and vitamins and fatty acids. So basically, there's no middleman between the food and your genes. So whatever you eat is directly communicating with your genes through those nutrients and minerals and vitamins. Another example of this is there was a study done in the 1930s by Fred Hale, who went to Texas Agricultural Experiment Station at College Station. And he was able to deprive pigs of vitamin A before conception so that the mother would produce a litter without eyeballs. And then the next litter he would give her enough vitamin A and the next litter would have eyeballs. So that's showing that the development of eyeballs was turned off only temporarily. There was no permanent mutation, only temporarily, by the lack of vitamin A in her diet. And basically how that happened, why would vitamin A affect the development of eyes, is that vitamin A is derived from retinoids, which come from plants, which in turn depend on sunlight. So in responding to the absence of vitamin A by turning off the genes to grow eyes, it is as if DNA interpreted the lack of vitamin A as a lack of light, or as a lightless environment in which eyes would be of no use. So these eyeless pigs had lids very much like blind cave salamanders, and it's possible that these and other blind cave dwellers have undergone a similar epigenetic modification of the genes controlling eye growth in response to low levels of vitamin A in a lightless, plantless cave environment. So that just shows that that's how our that's how our genes would modify to be better adapted for whatever environment that you're in. So excuse me. So basically, it's kind of like a software program that recognizes the environment outside of us and then reprograms our genes when we have children. So when it recopies them to our children, it programs our genes and says, this is most suited for the environment outside now. Which is why it's important to eat food that's close to the original source and least unprocessed, because otherwise the environment outside is very different from what is the actual reality of what's outside. That's the basics of how your genes and food are definitely connected, and how your food affects your genes, and how your genes will affect your children and even your grandchildren, because it's been shown that the maternal diet can affect offspring up into at least two generations. So whatever you eat now can affect how your grandchildren eat. So that's basically chapter 2 in a nutshell. In the next chapter, which is about symmetry and beauty in nature, I'm going to talk about more of Marquardt's mask, and how when we eat right, we basically will have beautiful children, because beauty implies function, and all of us are trying to find the most functional and healthy mates, which equates to beauty nowadays. and that no matter what race or anything like that, we all have the same ideal, because every person who is beautiful has the same basic skeletal geometry. And I'll explain more about that in the next video posts. I hope you guys enjoyed Chapter 2. It was really interesting, and the best thing that I can say you should do is definitely to read this book, because there's so much more that I'm not saying, even though I made a really long video again. Again, I hope things are well in the States, or wherever you are, and Italy is spectacular. Arrivederci.